Grazie. Grazie.
There was a contribution by Enzo Marinari and Giorgio Parisi in the early 90s about this kind of simulations. So I just will summarize briefly a little bit of the path since the beginning of the 90s to nowadays. So I will talk about models based on atomic models, so on atoms. Interaction between atoms. And in the early 90s appeared this need about extending the simulations, the methods, using theories coming from statistical mechanics. And this is actually an effort that is still going on. And it has been a little transformed in the early 2000s with the advent of metastatistics, metadynamics. And so I will use a little very simple model just to understand the things, but it's transferable to more complicated molecules. Our aim is to understand the exploration of the conformational landscape of heteropolymers like proteins, not only to understand the folding process, but mainly to find in the configurational landscape all the properties that can emerge from the sequence of the protein itself. So this is a very simple force field that we can use for a simple model of a macromolecule. And this is actually, because one thing that we have to acknowledge in the many schools in physics and also in chemistry, especially in Italy, was in the 90s the strong push towards the use of computational models or computers, because in the 90s there was the advent of, say, the workstation and the modern high-performance computing infrastructures that were somehow starting at that time, with the Cray supercomputers, for instance. And there was a transfer of a lot of knowledge in the field of statistical mechanics and also quantum chemistry to take profit of the advent of this huge computational power. For instance, this was our contribution in the early 90s, actually the late 90s, to use the hardware provided at that time by the high energy community to make molecular dynamics, but this is just a little. So I will briefly describe a little model where the interest is in describing through this interaction potential just a large-scale property that is the end-to-end distance in a polymer. So this is a well-known Biden-Spring model of polyethylene. And it is important, this simple model, because there are exact results coming from theory. And there is a landscape that is actually similar to what a complicated heteropolymer shows. So there is a folded conformation that is, in this case, the extended one, that has the lowest energy possible in the landscape. There are coiled conformations that are characterized, that we saw this morning, by the large degeneracy of energy in these states. And actually hidden in this bunch of structures, there are also structures that are interesting in terms of microscopic properties, but they are hidden in the bunch of high entropy configurations. So there is an exact formulation of the end-to-end distance that is called the characteristic ratio for this kind of polymer, and we made in the years we put a lot of effort in describing with molecular dynamics these exact results. So it took 30 years to reach this kind of level of accuracy. Molecular dynamics is a very powerful technique that emerged in the 90s, but it has a lot of limitations, and actually this was one of the great limitations. So the information added by Enzo Marinari and Parisi was actually to use the temperature as a dynamic variable in this kind of simulations. It was done in a Monte Carlo scheme, but now Monte Carlo and molecular dynamics has been combined together in order to have a lot of exotic way to do molecular simulations. For instance, this is a contribution for this year made by this, mainly by this student Masataka Yamauchi, who comes from a Japanese group by Isashio Komura. They are experts. They elaborated the idea of the stochastic combination of many replica exchanging temperature as a dynamic variable, and they elaborated the way in a very complicated way, in a way that the acceptance ratio of the exchange is almost always 1. So all moves, all the transfer of temperature between the replica are accepted. So the point is a complicated combination, a complicated choice of the way the temperature are exchanged, and in this way you can realize for different replica an exchange of temperature with other replica where the transfer is very efficient. This is a typical trajectory one can observe, where this is a replica exchange molecular dynamics. This is the method that is almost a routine method in high-performance computing simulations of macromolecules. So every day, 100 of high-performance infrastructures are running 100 of replica for, say, 10 days or 1 month about, obtaining trajectories like this. And you can see that this is the temperature of a replica, and so once the temperature is low, you see that the end-to-end distance, so the extension, so the accessibility of the lowest energy configuration becomes high. So this is a way to sample the transition between structure, the folded state that is in this place or in this place, towards unfolded states. And the idea, the goal of our simulations is to go back and forth from these states. And more exotic, more advanced methods allow this sampling better and better. As is shown here, so the method allows to compute exact values of the characteristic ratio for this simple system, but at a very high cost, because replica exchange, so this blue line, that is the routine method applied for macromolecules, takes, let's say, 20 nanoseconds for such a small system that is a polymer of 40 atoms, let's say 40 monomers. So I mean, there is a big problem in this kind of simulation, so they are very popular and people are using them. Sorry. I'm sorry, I just pushed the wrong button. I escaped. There are other methods, sorry, I will be very quick, but there are other methods that actually they came in the same age, so the early 90s. Actually, in the paper in the article by Enzo Marinari and Giorgio Parisi, actually at the end is mentioned that at the same time appeared another method that is called the multi-canonical method. So it's a way that also the temperature is a dynamical variable that is used to build some histograms of the energy, and this histogram is used to enhance the sampling of the conformational landscape. This is a sort of a little different method, and we tried to work with Berg, one of the inventors of this method. So you can see that this method is called generalized ensemble Monte Carlo, is able to reproduce the exact results. So this was found actually in 2003. Generalized ensemble methods, or like multi-canonical, were not very popular because they are a little complicated to use. And so replica exchange molecular dynamics is more popular because it's easier. However, this method was very promising. The problem in this method is that the use of energy is not sufficient to, it's not enough to use this kind of methods with molecular dynamics, because the energy absorbs also the springs, the contribution from springs. And so the essential dynamic is not well explored because most of the energy transferred from these histograms goes into the extension of the beads, of the springs connecting the beads, so the short-range part of the potential. And this is the problem that actually, I think, so far has never been overtaken. And what we made in the past was to propose, according also to the idea of metadynamics, that maybe is well known in the audience, but however, the replacement of energy with a structural collective variable that is more related to the change of function of the protein itself. So this is actually is published in this paper here, but the idea is to define a free energy that is a function not of the collective variable, but of the average of the collective variable. And there is actually a formulation of entropy that is based on the information that is used to bend the ground distribution that one has in a simulation towards the given average. So this is an informational entropy. So using this kind of methods, one can actually unfold a protein like this. This is a frataxin, is a protein involved in maturation of the mitochondria. It's actually related to the energy metabolism we heard this morning. There are somatic mutations like these indicated here that are responsible of the transformation of the cell in a cancer cell. So the switch from the normal function to a pathological function, to uncontrolled function. And there are many experiments in this field that try to probe the change of stability of the protein with the given point mutation, so somatic variant. And it is important to understand that the use of collective variables is actually very powerful in sampling unfolded configurations like this. So this variable just destroys the hydrogen bonds in this yellow part, that is the beta sheet, that is the scaffold of this protein. So it is possible to first measure a distribution that is a sort of metastatistical distribution of the collective variable, and then to change the distribution from the ordered state that is represented by the blue line, sort of a low temperature state, to the high temperature state that is this red line. And you can see that what is very important is that there is an overlap between the low energy and the high energy states that is maintained in the band metastatistics. And finally, this is the measure of the free energy change upon mutation in the thermal, in the melting experiment for the protein. And what we were very satisfied to distinguish between those variants that provide a positive change of free energy, that meaning that the protein becomes more stable than the wild type, towards most of the other variants that actually enhance the probability to unfold the protein. So the main message of this little contribution is that with the advent of computers, actually the school of theoretical physics also in other kind of other fields of science, especially in Italy, where theory was well supported because of many reasons. There was this success in combining ideas in theoretical physics with the power of computers. So let's say that the experience is made by computers. We can collect a lot of experiments in silico using computers, but it is important to use fundamental theories in order to give a soul to this kind of experience. Okay, thank you for...
Thank you very much for the timing, perfect timing, and for the interesting talk. Is there one question? So.
Un attimo.
Puoi parlare al microfono?
didn't explain this because it's too long. It is written in the article, but however, unfolding of frataxin, I don't know if you know that this protein binds iron, and there is a sort of acidic ridge in one of those alpha helices I showed in the picture. And there is a switch in the function of frataxin from loading iron, storing iron, and giving iron to those iron sulfur clusters that are part of the electron transport chain in the mitochondria. So actually, this is a question also for the talk of this morning. So it's not clear why these somatic mutations that impair the storage, in our opinion, the storage of iron, why these are related to cancer. But it is interesting that the mutation that enhances the stability of frataxin is in one particular type of cancer, that is cancer in liver cancer. The other types of mutations are in other kind of cancers. So it is something that we have to exploit, but of course this is out from the problem, because we can understand which is the reason of the increase or decrease of stability compared to wild type. Then the problem to attach this kind of interpretation to cancer is very complicated, because it deals with the compartment where the protein is and the partners which are with which the frataxin protein interacts. However, there is a nice complex now has been measured by cryo-electron microscopy, where this kind of mechanism of a transfer of iron from the storage to the iron sulfur cluster biogenesis has been a little explained. So we call this plasticity because the variants actually act on the way the protein can switch between these two functions. So actually, this is important because it's not only a folding problem. So you don't need just to understand which is the lowest energy structure, but you also need to understand which are the structures close to it in order to understand how the protein works, because it's plastic.
Thank you very much. I'm sure there are a lot of other questions, but the schedule is incredibly complicated, so we need to go fast. And kindly ask Marco Cosentino Lagomarsino from Milan to take the stage. Thank you very much. I think the scope of these talks is to give some stimuli, and then discussion can follow, because otherwise we cannot reach the end. Thank you.
Okay, well, can you hear me? Yes. So I'm really grateful to the organizers for having me here, in particular to Riccardo. My group in Milan is based on expertise in statistical physics, but physically we're based in a biology institute with iPhone. And as such, we're really grateful to Giorgio Parisi for opening the way to interdisciplinary endeavors such as ours. My subject for today will be this macroscopic or thermodynamic theory of biosynthesis. These kind of theories are an effort of many groups. Particularly, they started from the group of Terence Hua, whom you might know, in San Diego. And so we are just part of a wider effort. How do they work? And why are they called thermodynamic? Basically because they are theories of macroscopic fluxes. So you have, I think I have a pointer, but I don't know if it works. So you have two macroscopic fluxes. One is you have nutrients, and you have a flux making amino acids from nutrients. This is catabolism, metabolism. And then you have another flux using amino acids to make proteins. This is translation. Okay, so it sounds really simple. There are some key quantities, such as this ribosome allocation function. This is called, it's the fraction of ribosomes that are making ribosomes. So ribosomes are making everything else, but they also have to make themselves. So if you make too many ribosomes, you're wasting, you don't have enough to, you're not making enough of yourself. And then if you're making only ribosomes, you're not making yourself. So there is a trade-off. And you have to have a regulation to basically optimize this trade-off. And other important quantities are these fraction, let's say proteome partitioning. So the fraction of your proteome that is, in our case, for example, ribosomes, which is different from the fraction of ribosomes that are making ribosomes. And these quantities, for example, follow growth laws, such as the one that you see. You might see it's kind of out of focus in the lower right of the slide, which was found by Terry Hua and co-workers 10 years ago. So basically it tells you that this proteome composition doesn't really depend on details like where you're growing on, the nutrients that you're eating, but it's a generic function of growth rate. This is an astonishing, I think there's many of those. It's an astonishing finding from the complex systems viewpoint because it's very universal. So it holds for bacteria, a lot of species. Now it's been tested in a lot of species. And it holds for yeast, for example, which has an entirely different biosynthetic architecture. So it's interesting. So you can turn the principles that I told you about in equations, in dynamic equations for relating these fluxes, such as the ones that you see here. So the top equation is basically telling you that ribosomes are making everything, and it's relating the specific growth rate to the fraction of the proteome that is phi r, that is ribosomes, to the essentially the translation, the overall translation rate. And the second equation, which is relating this ribosome allocation to the ribosome, to the proteome partitioning, is basically telling that, is basically saying that ribosomes are making ribosomes. From these equations, you get some theories that have been studied, also out of steady growth, so out of balanced exponential growth. And for example, in nutrient shifts, which is basically what we will look at today. And, but these theories have a limitation, have two main limitations. The first limitation is that they don't really tell us how you set, how you read your nutrient levels and you set your ribosome allocation. And this is done mechanistically by the cell using some sensing and affecting circuits. And the other limitation, also related to the fact that most of the groups active in this area, they do bulk experiments, they don't do single cell experiments, is that these theories, they don't really tell you how the growth rate talks to the division rate. So in balanced exponential growth, the growth rate and the division rate have to match, but out of steady state, even the means, they don't have to match. So this workshop is, I was told, is about also enabling technologies. And in our case, the enabling technology is this one. It's microfluidic devices that allow you to look at cells one by one, dynamically, growing and dividing, and also proxies, in our case, of the proteome. And in our case, we will be looking at nutrient shifts, so going from one condition to another condition. From the physics viewpoint, it's kind of a response, very complicated response function of this system. You perturb it and you look at the relaxation. And then we will try to match with the quantitative model. So we have these two questions. One is about how do you compute, sense the nutrients and compute your growth rate and affect it, allocating your ribosomes. And the second one is how do you set division rate. So depending on how much time I waste, I might get through all my talk or just half of it. But let's start from the first question. So how do you get the resource allocation? If you ask a biologist, a bacterial physiologist, they will tell you, well, it's PPGPP. Okay, but what is PPGPP? It's just one in the G of the nucleotide with some phosphates and a sugar which is not, which is used both as a sensing and as an effector of the regulatory function that we are interested in. So how does it work? I will try to explain it in a crash course. PPGPP, through some proteins, is able to sense the nutrient levels through the uncharged tRNAs. You know that you have to charge the tRNAs in order to perform translation. So PPGPP is produced when the tRNAs are not charged. So if you are low in nutrients, you will have a lot of uncharged tRNAs, and this protein called relay will make PPGPP go up. Okay, then it's a sensor. But it's also an effector, because when PPGPP goes up, it sort of interacts with RNA polymerase, on specific promoters, the ribosomal promoters. So it can repress specifically the transcription at ribosomal promoters. So it's an effector of a regulatory function. Okay, this is just all together. And this is again, we have this data from shift experiments. In our case, we were adding this case. I will tell you another case later. Thanks to this collaboration with our friends in Cambridge, they were adding amino acids, and we were looking at these proxies for the ribosomal sector through some reporters, GFP reporters. So we can, with some work, get the macroscopic quantities that we're interested in. We can also get single-cell quantities, and I will tell you about those a little bit later, but there's a lot more work that we need to do. So as you can see, during the shift, the growth rate increases. The ribosome fraction, which is our phi r, the growth law quantity, overshoots a little bit, so it's a non-trivial response function. And also the biosynthetic rate and the ribosomal location, which we get through the balance equations that I described before, because with the data that we get are the first two. So in the current theories from this Terence Hua paper published in 2017, they don't have this regulatory function. And what do? They use steady data to infer it. So they say this chi-r is a function of nutrient availability through the basically the effective translation rate parameter. And we get it from steady state data. This is the relation that you see here. And we can test it with our data, whether during the shift we get this relationship, and we don't. So we really need the mechanistic theory that I'm telling you about. So that's what we do. I won't show you the equations, but we take their framework from Ericsson 2017, and we build in the PPGPP as a sensor, as an effector, and an effector of ribosomal location. And we also make, which is, so the first panel is this. We also add other ingredients that are necessary. One is the right panel, it's the specific amino acid dynamics. So the flux creates amino acids, which the nutrient flux, sorry, the catabolic flux, which are then consumed by translation. And the other ingredient assumption that we make is that ribosomes are partitioned randomly on transcripts. So they have equal probability to translate a ribosomal transcript and a non-ribosomal transcript. So it depends. So the chi-r will be the ratio of ribosomal transcripts to total transcripts. So this is results of our model, and we can see that these overshoots that are not found in current theories and also in current data, because they're using bulk data, are general in these models, which if you look at the eigenvalues always give us complex parts of damped oscillations. And I was surprised, some expert of this field, and I actually was thinking this way, might think that this is due to the transcriptional delay, because it takes you a while, once you read the nutrients, to partition your polymerases and make new ribosomes. So you have this transcriptional delay, but this is not responsible for the oscillations, because in the theory we can remove the transcriptional delay and we still have the oscillations. So how much time do I have left? Okay, so I'm rushing, but I'm still on time. We can get into the division rate. So first, so then now the question is, okay, we kind of have a theory which might not be, all-inclusive for a mechanistic regulatory allocation of ribosomes. But now the question is, okay, but these cells are also dividing. How do they decide their division rate? And the conventional wisdom, if you ask a biologist, is, okay, you have circuits that read by dilution, for example, read the growth rate, and then compute some division rate, which is some cell cycle proteins. But I will show you that this is not the case. They're done in parallel, and they're done through PPGPP. And the first evidence that I will show you, again, it's models and data, but it's based on this data where we decouple the system, thanks to this collaboration with Sander Tans and Greg Bokinski in the Netherlands. So they are able to increase through, like, again, some inducible genetic system, the levels of PPGPP tricking the cells in thinking that there are fewer nutrients, but there aren't fewer nutrients. There's just more PPGPP, and they look at the results in terms of growth rate and division rate. Okay, so what do you see when you do that? For example, you can look at the growth rate of these microfluidic experiments jointly with the added length, which is a proxy for the division rate. But it's actually more important than the division rate, as I will show you in a minute. And you see that the added length goes down much faster than the growth rate. So it cannot be reading the growth rate and computing something. These things are happening, the best case scenario at the same time. And we basically formulated the same question more stringently using mathematical models, and we tested two different models. One is the hierarchical model, where you have nutrient levels, growth rate, and then division rate. And one is the other model where, let's say, PPGPP is able to regulate both at the same time, both division rate and growth rate. And as you can see, only the second scenario can fit the data. There's something interesting, there's something else that's interesting about this data. I don't know if you noticed, but if you look at the green curve, which is the division rate, it actually increases before decreasing when you increase PPGPP, which means that they are accelerating before they slow down, and this tells us that the relevant variable is actually the added size and not the division rate itself. Somehow there's a circuit that measures the added size. So using the other data, we tried to make quantitative models of this possible division rate circuit. And now I'm switching data set again because this data set is much richer in terms of statistics, so we use this one rather than the other one. But this is just to show you that you see the same features. For example, if you look at the second, in this case you're going to a faster growth rate, and the second plot is the interdivision time. So you see that it has to decrease, but initially it increases, as the reverse as before. We tested several models. The details are not relevant, proposed in the literature for this division circuit, and they all don't fit this shift data, which means that these data are good for falsification of models, and it's very good when you can falsify models, because as you can see, there were a lot of models in the market. Then we tested another model that was proposed also in the literature, but with a variant. This model is basically a threshold accumulator model. There's a protein that's produced or something else that's produced at a volume-specific rate, constant, let's say, and then it has to reach a threshold number, okay. And then we could put in the theory that this protein could be ribosomal, for example. So it could be like a ribosomal sector protein or a catabolic sector protein. And we found that the data work very well if it's a catabolic sector protein. So the model is predictive if you assume that you have a threshold accumulation process that sets cell division, and that whatever is accumulating behaves like a C-sector protein. Okay, now I made a very long story short. I don't know if it was too short, but the main message that you should take home is the following. You have, so it's the one that the bacterial physiologists would tell you anyway, but then supported by models. PPGPP sets the growth rate, and at the same time, it sets division rate. Actually, it's not exactly what they would tell you, because the second part is less trivial for conventional wisdom. So thank you for your attention.
Very much. Thank you very much. And so if there's a very rapid question, because I gave you 2 minutes more than predicted.
Sorry,
so,
you should stop me.
No. I just want to apologize for the fact that we have such short talks. It's not not proportional to your scientific standing. It's just that we had constrained because this room can only contain a limited number of people, so this morning we could just have to close the registration, blah, blah, blah. Many reasons, so don't be formal and please don't feel offended. Any question, or we can move? Okay.
Is it the problem that the ribosomes are not uniformly distributed around the cell? You have
You mean the
proteins and
spatial
other proteins.
distribution of ribosomes? So this model doesn't assume anything on the spatial distribution. It's just a purely mean field model. But of course, we know that they are all around the nucleoid. There are super resolution data. But this doesn't really, nobody showed that this is actually limiting biosynthesis so far. So we don't know. There might be very fast growth conditions where it is.
Thank you very much again, Marco. So I kindly ask Marco to come up.
Okay. Thank you very much. Right, so my talk is going to be linked to at least a couple of other talks that we heard about this morning, and actually starts with this very, very often repeated, let's say, quote by Richard Feynman. And actually, my talk is, let's say, the more technical motivation of what we did is actually trying to understand what you have to do when you can't actually look at things. There's a lot of variables that you cannot look at, and still you want to have information about those. So what can you actually do in those cases? Now, the example I'm going to focus on in my talk is actually taken from bacterial growth. I'm going to talk about E. coli throughout the whole talk, but we've looked at other systems. E. coli is just, let's say, for the sake of, we have done much more work on E. coli than on any other system. So people have developed really, really effective experimental methods to track E. coli growth at single cell resolution by microfluidics. And they extract lots of distributions like these that basically represent, for example, interdivision times at single cell level measured across the entire E. coli population growing in different media. So the red one represents a very fast medium, and the whatever this color is like a very slow medium. Okay. Now, behind these results, there's a lot going on at metabolic level, as we heard this morning. So what one would really like to do is, besides having this information about single cells growth rates, let's call these macroscopic observables, one would also like to know something about what's going on inside, so the actual reaction rates, for example, the rates of metabolic reactions in cells. Now, we heard about how complex this is. What we didn't hear this morning is that there are actually methods, there's techniques that can actually probe metabolism. The most famous of these is actually mass spectrometry. I'm not going to go into the details of how this works. Let me just say this is a really, really complex analytical technique. It's not only an analytical method, there's also theory involved in that, there's inference involved in that, so it's really, really complicated. But the net result is that essentially if you do mass spectrometry, you take a population of E. coli cells in this case, you pass them through a spectrometer, the spectrometer crushes them, like literally, it's a destructive technique, and then it gives you an estimate of the flux through a small set of metabolic reactions, usually, let's say, up to 20, maybe 30, today somebody does up to 40, from the main carbon pathways of the cell. So I'm talking about glycolysis, respiration, maybe pentose phosphate pathway. Yeah, this is basically what you can cover. Now, this number here, even if you go up to 50, it's really, really tiny compared to the whole network. So this technique gives you information about a really small part of the network. The question is, can we get information about the other reactions, those that we cannot access with this technique? Now, let me just mention the fact that there are some intrinsic limitations to mass spectrometry that make it really, really hard to go far beyond this thing, okay? Now, there are proposals for improvements of this technique, but it's probably going to take whole new experimental setup to be able to overcome this kind of limitation here. So having a way, let's say, a different way to understand or to predict the other fluxes is, at this point at least, really, really important. So the way we do this is by statistical inference, okay? And just to give you an idea, I'm not really going through the details of this, but the idea here is the following, that as a picture, as a cartoon, what you should have in mind is that when people measure these kind of distributions of interdivision times or of growth rates or whatever, what they're really doing, they're looking at, oops, what have I done? Okay. So they're looking at the marginal distribution of a high dimensional distribution that represents the entire population using all the possible variables that can be regulated inside a cell. So you have to imagine that there is some really, really complicated and high-dimensional phase space of the system, so a set that where every point represents a possible state of a cell, and then a population is just represented by a bunch of points selected from these sets. So when people look at measure these distributions, what they are doing is actually looking at a low-dimensional sample from a high-dimensional distribution. Now, what we want to do is to use these low-dimensional samples to reconstruct the entire distribution, basically. If we can do that, then we get the information about all fluxes, for example, in metabolism, all metabolic fluxes, including those that you cannot measure. Now, in physics, people have been solving these kind of problems for a long time, and some of what Remy said this morning has a lot to do with this. The field is so mature that there is a fantastic review article about this written by somebody who's sitting over there. And it's been used in physics to study, for example, magnetic systems, in neuroscience to reconstruct neural connections, and in a bunch of other cases it's been used. So we didn't have to invent anything, we just had to apply it in a different way and to a different problem. So just to give you a really snapshot idea of what this means in practice, what you do when you, in order to reconstruct this high-dimensional distribution, is you solve a maximum entropy inference problem. So you're looking for the distribution of metabolic fluxes, in this case, that has the largest entropy and which satisfies, let's say, minimal constraints like normalization. And the other constraints that you want to be satisfied is that the means of the individual fluxes that you measure have to match the empirical values, okay? So there's like 20 or 30 constraints like this that you take from experiments. And this problem is actually really straightforward to solve on paper, and the probability distribution that actually gives you the best guess for the real high-dimensional distribution that you don't know and that you want to find has actually this exponential or Boltzmann-like form, where in the fluxes that are experimentally accessible enter in the argument of the exponentials multiplied by these constants here that are basically Lagrange multipliers, and they are essentially akin to inverse temperatures in statistical mechanics. So this is just the same thing that some of you have seen hundreds of times in physics, except that we don't only have one temperature, so to speak, but we have one temperature per each measured flux. Now, the hard part is not so much solving this problem, but actually finding the values of these coefficients that will reproduce the empirical means, okay? But if you're capable of doing that, and actually, if you read these articles, there's a bunch of techniques that will teach you how to do it, then you can actually learn the entire distribution. Okay, so, just to give you, again, a quick idea of the kind of results that we obtain, we can compute predictions for the fluxes that are not experimentally accessible, and they look like something like this, except that we have a bunch more of these distributions. And then, obviously, from this data, we can learn a lot about how the usage of metabolic pathways fluctuates from cell to cell, something that you don't see from mass spectrometry data, because it doesn't give you information about the whole distribution, but only about the means. Okay, so there's a lot to learn from this. I'm going to skip out this entirely to focus on something else. So the first thing is just to give you an idea of how different the results that you obtain from these methods are from those that you obtain from more standard techniques like FBA, for those of you who know what I'm talking about. So in these two panels, I'm reporting a couple of, let's say, macroscopic observables. On top, there's the acetate excretion flux for E. coli, and the bottom is the biomass synthesis flux for E. coli that are measured independently of mass spectrometry, right? And that we use as a reality check, so to speak. So we want to be sure that we're doing the right thing. And you see that how predictions and experiments compare. There's a few more that we can check. It always works very well. But to give you an idea of how FBA, of the predictions of FBA that you will obtain on these data sets, in this case, FBA would predict 0, while for this other variable, FBA would predict 1 throughout the entire range of growth rates for which we had data. Okay. Now, I'm coming to the thing that I'm personally more interested in, which is the following. So the data that we used were actually different experimental populations covering a broad range of values of growth rates, so roughly from 0.1 per hour to 1 per hour. And the way these experimentalists, by the way, was a group of Sauer in Zurich, where basically modulating growth was by changing the amount of carbon in the medium. Okay, so we were actually expecting that if these bacteria, if these populations were actually maximizing their growth rate, one would expect this inferred distribution to have a very large, at least the argument of the exponential, to have a very large projection in the direction of the biomass flux, because that would mean that the population is actually trying to maximize the biomass. Well, if you try to see how large the projection in the direction of biomass is, well, we cannot probably read out from this plot, but it's not very large. And actually, the projectome behaves very erratically. Sometimes you have very, very fast populations that have a very small projection in the direction of the biomass. So we try to understand or derive, let's say, a principle that is different from growth maximization in order to explain this data. Okay, and we were inspired by information theory. There's a very, very famous branch of information theory that is called rate distortion theory. And in rate distortion theory, what you do is you link a measure of the performance of the system. In this case, it's the distortion that let's say a receiver sees once you transmit a message along a noisy channel, to a measure of how noisy is the signal that you transmit, that the sender transmits along this channel. Okay? And the idea of rate distortion theory is that if you want your receiver to receive a message with a certain distortion, let's say a low level of distortion, then you have to transmit a message that has at least a certain amount of redundancy. Okay? Otherwise, the channel will corrupt the message so much that it will be impossible to retrieve it in the end. Okay, so we thought that basically in growing cell populations, there's a similar trade-off going on. So on one hand, cells definitely want to grow as fast as possible, but on the other hand, because there are fluctuations and you cannot get rid of the fluctuations, this also limits the performance of the cell, of the population. The population simply cannot grow as fast as it would like because of these fluctuations. I don't call it noise because I don't actually think this is noise or only noise. There's a lot of different sources of variance that go into that. But the nice thing here, is that you can actually link the mean value of the growth rate distribution, which is actually a very, very good proxy for the population growth rate, with the entropy of the distribution, of the high-dimensional distribution that you infer, or actually with the information content of the distribution, which is essentially minus the entropy. This is just technicalities. And so you can derive a mathematical bound that represents, it's actually this green line here, that represents, let's say, how fast a population can grow if it encodes a certain amount of information in this high-dimensional distribution that we infer, okay? So the green line is the best that you can do. And then because we infer the high-dimensional distribution, we can actually see how much information and how much, and what is the mean value of each of these distributions that we infer. And we can actually place points on this graph that represents real populations, and this is what you get. So what you see is that actually, these are, as you go from blue to red, the growth medium gets faster and faster, so the population grow faster and faster, and they actually approach the optimal performance, but they stay consistently below it. So they don't actually maximize the growth rate. They are, they're trying to basically implement a kind of trade-off between how fast they can grow and how diverse they are. They also want to keep diversity as large as possible. So, of course, this is a principle that, let's say, we kind of used to construct different models of metabolism that use this kind of idea to predict fluxes, let's say, without making FPA-like assumptions. And actually, I don't have time to go into this, but basically they also work very, very well in predicting fluxes, just, let's say, from scratch. So these are my conclusions, and I have to thank Anna Montoni, who actually did most of the heavy lifting here. And thanks for listening, and thanks for having me.
Thank you very much, very interesting. So, one quick question, yes, Marco, very quick. Let me remind you that everything will be streamed, so people will have the occasion to listen to your talks.
So are there mutants that actually do not have, or where you basically see that they get closer to the optimum, so they don't want to be diverse?
Sorry?
Is there any mutant that you can think of where you can actually delete this trade-off? And
yes, yeah, but we don't have a lot of data that we can use with the same high resolution that we have for this. And the other thing is, of course, we're looking at cancer for this kind of understand diversity and see, basically trying to trace how diversely cells respond to treatment with how diversely they use metabolic pathways, essentially.
Thank you.
Thank you very much, Andrea. Okay, so now we are really, our pace is very high. So Federica, please, it's from the CNR, and it comes. Okay, so please.
Okay. Okay. Good afternoon. My name is Federica Conti from the Institute of System Analysis and Computer Science of CNR of Rome. First of all, I would like to thank the organizer of this workshop for giving me the opportunity to present my work today. In this talk, I'm going to present you a new paradigm of medicine called network medicine and its application to breast cancer subtypes. So, let's start. Conventional medicine is based on the paradigm one gene, one drug, one disease. This means that it applies a reductionist approach mainly based on finding a single magic bullet, no matter the complexity of the phenotype, and focused on treating the symptoms, then discovering the causes of the disease. According to conventional medicine, also the definition of disease is mainly based on the organ affected by the disease, for example, lung cancer or cardiac disorder, or on the name of first discovered, for example, Alzheimer's disease or Parkinson's disease, or on the symptoms. Network medicine instead represents a real revolution in medicine because it applies tools and concepts of network science for identifying, preventing, and treating disease. In this new vision, diseases are rarely caused by a single gene mutation, but more typically by the deregulation of a network of interacting genes. And understanding the effect of this interaction can lead to the identification of new molecular biomarker and therapeutic target. Moreover, the integrated network of all physical interaction within the cell is called the interactome, can be interpreted as a map and disease as local perturbation. In this map, the molecular determinants associated with a given disease, called disease genes, are not randomly scattered, but they form localized subnetwork called disease modules. And these modules may overlap between similar diseases, suggesting that different diseases may share common molecular mechanisms. Thus, the slogan of network medicine is think globally, act locally. Following this paradigm, Barabasi and co-authors built the so-called diseasome, that is a bipartite graph consisting of two sets of nodes. One set corresponds to all disease, all genetic known disease. The other set corresponds to all known disease genes. And in this network, a gene and a disease are linked if mutations in that genes are involved in that disorder. The size of disease is proportional to the number of disease genes associated to that disorder, and the nodes are colored according to class to which they belong. The clustering of nodes of similar colors suggests that similar pathophenotypes have a higher probability to share disease genes than do pathophenotypes belonging to different disorder classes. For example, in this sketch, obesity here is more closer to diabetes than to cancer, in accordance with the knowledge that obesity is a risk factor for diabetes. Recently, in my lab, we have developed a new network-based approach called SWIM that exploit the gene co-expression network to identify new disease genes associated to specific disease based on their gene expression profile. These particular genes were called SWI genes because they may have a key role in the switch or transition between two biological conditions, like, for example, from physiological to pathological condition. And the SWI methodology is freely available also in our language at this GitHub link. The SWI genes are very important because they have very peculiar topological features. Indeed, they show coherent patterns of co-regulation, suggesting that they may be co-regulated or functionally related. They form localized subnetwork in the correlation network, but they are not localable within their module, but they are important connectors able to convey information between different modules. For example, in this sketch, the orange node is a local hub because it interacts mainly within its module. The red nodes is a peripheral node because it interacts few in general and only within its module, whereas the red nodes is a SWI genes because it interacts both intra and intra modules, but it is not a local hub. In this recent paper, in collaboration with Professor Lo Scalzo of Harvard Medical School, we studied the interplay between SWI genes and human disease in the human interactome. In particular, we used a network-based separation measure, here defined, to assess the topological proximity between the SWI-based disease modules. In a few words, negative value of this measure indicates overlapping modules, while positive values of this separation measure correspond to non-overlapping modules. We studied a total of 14 disorders, including 10 tumor types, 2 cardiac disorders, 1 neurodegenerative disease that is Alzheimer's disease, and 1 pulmonary disease that is COPD. We found that the SWI genes associated to each disease are not randomly scattered, but they they are agglomerated in specific regions of the interactome. And this module overlap among similar diseases like cancer and cardiac disorders, but are situated in network neighborhood different for pathologically different disorders like Alzheimer's and COPD. In this way, we were able to build the first SWIM-based human disease network, that is a network in which nodes are the disease, and the link between two diseases occurs if they share a substantial number of switch genes. Also, in this case, the size of node is proportional to the number of the switch genes, and the nodes are colored according to a specific disorder class. In this network, labeled node, of course, correspond to the 14 diseases that we have studied, whereas unlabeled nodes correspond to other diseases that can be investigated in the future. This type of approach can be applied to study not only different diseases, but also different subtypes of the same disease, because as probably you know, the subtypes of the same disease may have very different clinical pathological features. In particular, in this other very recent work, we apply SWIM to different breast cancer subtypes available from the Cancer Genome Atlas database, in order to find a common molecular mechanism underlying to the different breast cancer subtypes in the framework of network medicine. In particular, by exploiting the information of the TCGA, we stratified the patient according to different classifications. A genetic classification, also known PAM50, so-called because it is based on the gene expression of a panel of 50 genes, and the immunohistochemical classification that is based on the presence or absence of 3 receptors on cell surface. We ran SWIM for each breast cancer subtypes of PAM50 classification, and in particular we compared the total of about 100 normal samples against more than 50 tumor samples for each breast cancer subtypes, and we obtained a total of switch genes ranging from 200 to 300, all upregulated in tumor condition. Then we mapped these switch genes on the human interactome, and we found that they form localized subnetworks, suggesting that they may be proposed as new molecular determinants associated to these subtypes. Interestingly, these modules overlap. Indeed, we found always negative value for the separation measure, suggesting that a pathobiological similarity may exist among these different subtypes. Similar results were found also for the other classification. So, then, searching for common switch genes of all breast cancer subtypes of both patients' classification, we identify a unique common molecular gene signature composed of 11 switch genes. Interestingly, these switches are involved in a cell cycle, in particular in the G2M transition. And among these 11, we choose to focus on the ORCA kinase because it is a kinase with a key role in cell growth and cell cycle progression. It has a critical role in mitotic events like spindle assembly and chromosomal segregation. It is deregulated in many cancers, and it cooperates with a lot of very well-known tumor suppressors like P53, and it is commonly used as a pharmaceutical target for the treatment of different types of cancer. The key role of ORCA protein in the framework breast cancer subtypes was experimentally validated through in vitro experiments. In particular, we found that ORCA protein was always overexpressed in all breast cancer subtypes, and that its inhibition led to reduce the cell growth and to arrest the cell cycle. Moreover, we performed also ex vivo experiment by analyzing different breast specimens, and also in this case we found that ORCA protein was overexpressed in all tumor tissues for the different subtypes. So, coming back to the diseasome drawn by Barabasi, thanks to SWIM, we were able to add a new molecular determinant associated to all breast cancer subtypes, that is ORCA, and applying SWIM to other solid tumors available from the TCGA, we found that ORCA is a switch gene also for lung cancer and blood cancer. And from literature, it is involved also in other important tumors like prostate cancer, thyroid carcinoma, or colon cancer. And then, in conclusion, I told you that, of course, network medicine represents a new era in medicine because it proposes a holistic approach that views disease as a complex network of interacting genes and not more as independent entities. In this regard, SWIM exploits network theory to identify the molecular determinant associated to a given disease based on their gene expression profile, and in particular, by applying SWIM to different breast cancer subtypes, we were able to identify a common mechanism associated with the ORCA pathway altered in all breast cancer subtypes. So, following the network medicine philosophy, we may suggest that targeting ORCA pathway could pave the way for novel therapeutic strategies that are no more organ or symptom specific, but rather mechanism specific. I would like to conclude my talk by thanking all the people who took part to this work, and of course, thank you for your attention.
Federica, thank you very much. I'm checking that we are accumulating delay, so if there is a question, it should be very rapid. Sorry for that, but you have many, many talks, 13 talks. So. A rapid one.
I missed the estrogen receptor as a connector gene. You don't seem to have it as such. Estrogen receptor, breast cancer, I think that should be one of the dominant connector genes.
Please answer to the microphone.
Yes, we focus on the ORCA, but there is a connectors that interacting with them. And in this case, we can explore the network, the module of the genes around this pathway. And this is one of the aim of the experimental group that work with us. This is the next step, of course.
Thank you very much. Thank you. So, she's from Rome. Also, in your case, the title is too long for me to, to pronounce it. So please just go ahead. Thank you very much.
Good afternoon, I am Valeria Becci, a PhD student from Università Cattolica del Sacro Cuore, and today I will talk about targeting of low non-coding RNA H19 cell adhesion molecule circuitry by EpiDrug, a novel tools to inhibit tumor growth and metastasis in prostate cancer. Prostate cancer is the one of the most common cancer in West country, and it's an androgen-dependent tumor. Nowadays, aging is the only recognized risk factor correlated to the decline to androgen in estrogen ratio. Radical prostatectomy and radiation therapy represent the standard treatment. However, 30% of patients develop a biochemical recurrence, creating the need for additional therapy and new early biomarker. A previous transcriptomic analysis on prostate cancer-derived primary culture led to identify a prognostic transcriptional signature. This transcriptional program is driven by estrogen and hypoxia signaling and regulates the expression of different genes, including low non-coding RNA. We focus our attention on H19, one of the most sensible low non-coding to estrogen and hypoxia. In general, low non-coding acts as a key modulator of both physiological and pathological cellular processes, and H19, starting from early stage, has a role in tumorigenesis and metastatic progression. Estrogen and hypoxia act as pro-tumoral stimuli, inducing invasion properties. Here, we observe an induction of metastatic properties in aggressive primary prostate cancer cell. In this particular condition, estrogen plus hypoxia, we observe a peculiar H19 downregulation associated with upregulation of its transcriptional target genes, in particular a cadherin and beta-integrin subunit. This mechanism creates a cluster of cells that move together, creating the so-called cohesive metastatic phenotype or collective migration. Based on the results just discussed, the effect of epidrug currently used in different human clinical trials and in vivo model was evaluated. Among these, we selected JSK-J4 for its ability to interfere with H19-dependent transcription. Indeed, we observe a reduction of cell adhesion molecules induced by the combined treatment, estrogen and hypoxia. Data confirmed by the reduction of metastatic properties. Our working hypothesis is that interfering of H19 cell adhesion molecules circuitry may affect in vivo key mechanisms driving tumor invasiveness. The present project aims are to test in vivo efficacy of epidrug in prostate cancer tumor mouse model, following to analyze epidrug effect on preclinical prostate cancer model, in particular in ex vivo organotypic slide culture, and finally, to identify at the time of biopsy prostate cancer patient potentially targeted by epidrug treatment. Starting from this point, PC3 luciferase positive cells were selected as experimental model for in vivo study. The cells were stably silenced for H19, and then we observe an upregulation of cell adhesion molecules. Parallel, we observe also the increase of metastatic properties. Also, in this case, JSK-J4 reduce the level of the basal level of E-cadherin and beta-4 integrin. Data confirmed by the reduction of invasiveness. Then we move on to in vivo experiment. Xenograft mouse model was generated through subcutaneous injection of cell into the node-shed mice, immunodeficient mice. As I showed, silenced H19 cells show a higher growth rate respect to vector cells, and J4 reduced the tumor volume. So J4 acts also in vivo. In human prostate cancer, metastasis can spread in different sites, and lung, liver, spinal, and bone represent the most common site. We are able to reproduce human metastatic dissemination by injecting our cells into the tail vein of NGS mice. As you can see, both cells, both silenced H19 and vector cell metastatize to the lung. But the immunohistochemistry revealed that silenced H19 cell create a cluster of human cell into the alveolus as well as in blood vessel, while vector cell appeared as individual or single cell. Regarding J4 treatment, we observe a reduction of metastatic formation, and silenced H19 cells show a more number of metastases. Similar results were obtained in liver, and where the higher diameters induced by silenced H19 cells was reduced by J4 treatment. Of the note, we obtain also bone metastasis. As you can see, this transversal section of spinal column, the cells create a cluster into the spinal column, and here we observe an invasion of both bone matrix and cartilage. Then we selected a prostate cancer-derived organotypic slide culture as ex vivo preclinical model. These were obtained from fresh surgical explant of prostate cancer tumor, and this model summarizes the peculiar characteristic of original tissue. Here we observe that after G4 treatment, we have a significant reduction of E-cadherin and beta-4 integrin in about 60%. Parallel, we have an induction of cell death. These results are in according to data obtained in prostate cancer cell line and mouse tumor model. Another line of research is aimed to identify at the time of biopsy of prostate cancer patient with altered H19 cell adhesion molecule circuitry and consequently targetable by epidrug. We analyze protein level by immunohistochemistry and RNA level by digital Doppler PCR of H19 cell adhesion molecules circuitry biomarker in a small cohort of prostate cancer biopsy. In this pilot study, we have a group of recurrent patients with progression disease after surgery. Univariate analysis of a selective biomarker show that only eNOS is significantly associated with the recurrence, while beta-4 integrin show a trend. The other standard clinical and histopathological parameters like biotic Gleason score or pathological on PSA are not correlated with progression disease. Then, through a multivariate analysis, we define a bioscore. Bioscore is defined by the combination of selective biomarker, in particular defined by the higher expression of eNOS, If2 alpha, beta-4 integrin, and E-cadherin, and lower H19. The presence at least 3 out of 5 of this selective biomarker is significantly associated with the recurrence and progression-free survival. Then we can conclude that GSK-J4 epidrug negatively affect in vivo and ex vivo H19 cell adhesion molecules circuitry, thus reducing tumor growth and metastatic dissemination. It's possible to identify at the time of biopsy prostate cancer patient with altered this circuitry. And this represents a possible new target therapy with potential impact on prostate cancer clinical management. Thank you for your attention, and thanks all my colleagues.
Thank you very much. Any question from the audience? You might receive some questions on YouTube, so please, thank you very much for your talk. Thank you. So Pasquale Palumbo, it's you. Grazie. So Pasquale Palumbo from Milano Bicocca, and he's going to talk about yeast metabolism. Please.
Thank you. Thank you, Mr. Chairman. Good afternoon. Today I'm going to show you some results, also some recent results on mathematical modeling, multi-scale integration mathematical modeling. This is a joint work. Marco Vanoni, Lilia Alberghina, Stefano Busti and myself come from the University of Milano Bicocca. Federico Papa is from the Italian National Research Council, and this is work funded by SISBio, the ISBE Research Infrastructure. Okay, so somehow everything started from the search for whole cell models, since recent studies underline the need to develop quantitative whole-cell models, so predictive whole-cell models to explain the complex phenotypes and to accelerate biological discovery. So there are many challenges, of course, in this kind of research because of different time scales, since you have to investigate different kind of biological functions, so different layers, so we need hierarchical modeling, different levels of graining to be explained by means of different kind of models, and of course also different kind of data. So we may have steady-state data or time-varying measurements, interactomics, metabolomics, proteomics, and so on. Everything may be said that started from this seminal paper from Jonathan Carr et al. In 2012, where he first successfully developed a whole-cell model for Mycoplasma genitalium, and it could replicate a lot of biological data, but it was a very basic microorganism, so it was a sort of starting point, since it was a bacterium, just about 500 genes, so no signaling pathways, no adaptation to external cues, and so many other simplification because of the kind of animal model chosen. On the other hand, since 2012, many research groups started to look for a whole-cell model for eukaryotic cells. And my research group worked on Saccharomyces cerevisiae. It is an animal model for eukaryotic cells, and it is much more complicated since, for instance, it has a different compartmentalized cellular organization, a genome which is tenfold larger, different signaling pathways, a large set of homeostatic response to external cues, and a sophisticated nutritional modulated sensing. So there is much more than the bacterium considered in 2012. Okay, as it was previously said by Marco Vanoni, this talk is strictly constrained to his. Indeed, this is the coarse grain model replicating metabolism, growth, and cycle. And of course, we can apply the building of the whole-cell model by means of different cellular modules. So, for instance, here we have the module for metabolism and growth, here the module for growth and cycle, and of course the interconnection is extremely important. So in my talk, I will talk about the possibility to exploit this model as a backbone according to which we may blow up different modules. So for instance, we are presently working on a molecular module to be plugged in and dealing with ribosome assembly. And we have started by taking into account the large and the small subunit and the molecular details, and as previously said, we have developed a different kind of models according to a different level of granularity that is required. Another work in progress is to exploit an already existing model, an already existing molecular model from Brummer et al. This is the work by Thomas Hoefer and Lydia Alberghini in 2010, dealing with the initiation of DNA replication. So we take an already existing model from the literature, molecular model from the literature, and we aim at plugging it into our coarse grain model. So also in this work, a pivotal role was played by multi-site phosphorylation mechanism in order to control the initiation, the coherent initiation of DNA replication. In my present talk, actually in the rest of the talk, I will give you some detail of another kind of molecular modules already published by my research group, which deals with the G1S transition module. So at first, a couple of slides to tell you what it predicts, what it represents, and then how it could be exploited as a plugin from the coarse grain model. Okay, so what is the G1S transition? Well, the G1S transition is the pathway to express the genes of the G1S regulon. So these genes are controlled by a couple of transcription factors, which are SBF and MBF. The way a cell is able to temporally schedule the G1S transition is by means of an inhibitor. Actually, there are many inhibitors, but the one which plays a pivotal role is Wi-Fi, which binds onto these two transcription factors, and so somehow prevents their active activity on the expression of the G1S regulon. So the whole G1 phase may be thought as the way these transcription factors get rid of the inhibitor. In this model, many feedback mechanisms are involved. For instance, the first genes to be expressed are those of cyclins 1 and 2, which play as controllers themselves to get rid of Wi-Fi. So this is an important feedback. And so at the end, most of the genes are expressed and DNA starts. So this molecular mathematical model aims at describing this machinery. The whole story may be synthesized as a sort of sensing plus setting, in the sense that the cell, and this has already been said by Marco Vanoni before, somehow senses the size at a given time instant, and the cell wants to reach a critical cell size before to start the DNA replication. So sensing is the way a cell realizes the size at a given time instant, and setting is the mechanism according to which different nutrients, different metabolic conditions may vary the amount of this critical cell size. Besides, the G1 phase may be divided into two sub-periods, namely T1 and T2. One period, T1, is a so-called noisy period because it varies in size according to different initial conditions. So, for instance, think about different initial sizes. And it works as a sizer, so this is the reason for we also talk about sizer plus timer. And the way T1 modulates its size according to the initial or to the present cell size is actuated by means of Wi-Fi phosphorylation. Wi-Fi was the inhibitor. From a mathematical viewpoint, our model is, of course, complex. Otherwise, we could not be here in a workshop of complex systems. We have continuous and discrete states. Continuous because some state variables evolve continuously in time because they are concentrations of proteins or of ribosomes or other molecules. And to this end, we exploit nonlinear order differential equation or according to standard or non-standard mass action laws. On the other hand, our model deals also with discrete states because, for instance, a gene may be active or non-active, may be expressing or not expressing, or because of different level of phosphorylation states. And these discrete states evolve according to a master equation, so they evolve in a stochastic manner. Just to get you in the flavor of the mechanism, here we have a set of genes and this is one of the transcription factors, and so these genes starts to be expressed when this transcription factor binds to them, and they, for instance, express for 136 molecules. The problem is that SBF does not bind really for work, but it binds inhibited by Wi-Fi. So the cell needs to get rid of Wi-Fi. How does this happen? Here, these points, white circles here and here, are phosphorylation sites. SBF needs to have the 4 sites phosphorylated to get rid of Y5, or Y5 needs to have these 4 sites phosphorylated to release SBF. The point is that we also have these 8 other phosphorylation sites that simply do not work to release Y5. So they are decoy sites, and they anyway can be occupied by the CDKs that, by the enzymes that work to make phosphorylation. So everything may happen. At the end, for instance, here we have found these 4 sites phosphorylating, these other sites phosphorylated, so this happens first, then Y5 goes away and the expression starts. So this is the reason for we have these kind of discrete states, because we have 1 phosphorylation, 2 phosphorylation, up to 12 possible phosphorylation. Yes. Some results. Well, in this picture, I show the way the genes are activated in time according to different setting of the phosphorylation sites. This is interesting because we can apply our model to think about what Albert Einstein called the thought experiments. We know that we have 4 regular phosphorylation over 12, but what if we had 5 over 12 or 8 over 12 or 1 over 12 or any other kind of possibility? I go fast. Another very interesting result was that our model suggested new experiments. Actually, it had been the reviewer to suggest us to use the model to suggest new experiments. But in any case, it really worked well because we implemented a new mutant according to what our model suggested, and we had very interesting and coherent results from experiments and the model. And finally, the most important point of my talk, we exploited this molecular module inside the coarse grain model. So here you see here you see in each panel 3 curves. They are 3 protein distribution. One is from experiments, the other is from the coarse grain model without the molecular module, and the third is the coarse grain model where the timer dealing with the G1S transition was substituted by the molecular module. And so we set the system in order to reproduce faithfully the previous experiments, and of course it could provide many other possibilities because we could make, we can make a different kind of theoretical mutation according to the G1S transition module. Here I have reported the results from glucose 2%, here glucose 0.05%. This was a mutant implemented by the module, and of course we could show many other results. So in conclusions, our research investigates the building of the whole cell models. Of course, we are in a good position, but we still have to work to deal with the other modules that I mentioned before. We exploited the Saccharomyces cerevisiae as an animal model. The approach consists in a of a multi-scale integration of metabolism, growth, and cycle, that is the model described by Marco Vanoni before. First results show the proof of principle for the G1S transition, and there is a work in progress for the ribosome assembly and the initiation of DNA replication. So that's it.
Thank you very much, Pasquale. We are actually 2 minutes late for this talk, so I don't know if there is a super urgent question. Otherwise, I would go to the next talk because we still have 5 talks to go, and we can continue talking after the session. So we are going back to some clinical research with Sara De Martino. So thank you very much.
Good afternoon, I am Sara De Martino, a PhD student from Università Cattolica del Sacro Cuore, and today we'll talk about metabolic rewiring by long-encoding RNA MALAT1 targeting in prostate cancer, a multi-pronged approach. Prostate cancer is a major cause of cancer-related death in men worldwide, mostly in Western countries. And it is an androgen-dependent tumor. Therefore, therapies targeting the androgen receptor signaling are first-line treatment, but resistance often occurs, leading to castration-resistant prostate cancer. It is characterized by maintenance or reactivation of androgen receptor signaling, by amplification, mutation, formation of variants, or aberrant activation of androgen receptor. Prostate is a unique and peculiar metabolism that changes during tumor progression. In normal prostate, OXPHOS and TCA cycle are inhibited with citrate secretion as a component of seminal liquid. In prostate primary tumor, OXPHOS and Krebs cycle are restored for an efficient energy metabolism with oxidation of citrate in order to produce more ATP. Only in metastasis, we observe a high glycolytic rate and inhibition of OXPHOS and TCA cycle, and this is called Warburg effect. It is well known that lipid play a key role in prostate cancer development and progression. So another feature of prostate cancer is the so-called cholinic phenotype, characterized by increased level of phosphocholine, overexpression of choline kinase A. Phosphocholine is the first intermediate of Kennedy pathway that leads to the synthesis of phosphatidylcholine, an essential phospholipid for biogenesis of plasma membrane. Choline kinase A is an enzyme responsible for the conversion of choline into phosphocholine. This aberrant lipid metabolism leads to the regulated cell proliferation, invasion, metastasis, and development of drug resistance. The high level of choline and phosphocholine are biomarkers used in PET-CT as tracers to detect local and distant metastases. For this reason, choline kinase A is a biomarker for metastasis and recurrent prostate cancer. The explosion of novel RNA classes, for example, non-coding RNA, has raised the possibility of alternative therapeutic strategies that mimic or antagonize the function of transcripts. In particular, antisense RNA promotes degradation of mature RNA. Moreover, long-non-coding RNA acts in gene regulation and in several biological processes. Therefore, long-non-coding RNA have been associated with a significant number of cancers, suggesting their role in different hallmarks of cancer. I focus my attention on MALAT1 role in metabolism reprogramming. MALAT1 is highly expressed in a variety of cancers, including prostate cancer, which has been reported to be involved in tumorigenesis and tumor progression. For this reason, MALAT1 could be a possible target for RNA-based therapeutics. So the aim of my work was to achieve a metabolic rewiring by MALAT1 targeting as a novel RNA-based therapeutics in prostate cancer. I focus my attention on energy metabolism, in particular glycolysis, TCA cycle, and OXPHOS, and in lipid metabolism, in particular phosphatidylcholine and sphingomyelin synthesis. These bioactive phospholipids are essential for plasma membrane biogenesis. I use as a tool to targeting MALAT1 gapmers. Gapmers are short antisense oligonucleotide that binds the target RNA, in this case MALAT1, promoting its degradation. I use this approach in vitro model using prostate cancer cell line, in which the depletion is efficient and stable over the time with anti-proliferative and pro-apoptotic effect. As an ex vivo model, I use organotypic slice culture obtained from surgical fresh explant from prostate tumor. Also here we observe a good downregulation, about 40%. Transcriptomic data upon MALAT1 targeting using different prostate cancer cells show upregulation of gene involved in RNA synthesis, transcription, and apoptosis, and downregulation of gene involved in cell cycle, protein synthesis, and metabolism. Transcriptomic data prompt us to evaluate energy metabolism rewiring upon MALAT1 depletion by gapmer. We observe a high glycolytic rate and a low level of TCA cycle, as demonstrated by the production of lactate and the downregulation of malic enzyme, as an example. The low level of TCA cycle leads to the secretion of citrate. This is important because citrate is a differentiation marker in normal prostate, and we observe this secretion of citrate in PC3 cell line, the most undifferentiated metastatic prostate cancer cell line. We move also the block, the inhibition of OXPHOS, strengthened the hypothesis that in prostate cancer cell, MALAT1 depletion restore a basal metabolism similar to a normal prostate epithelial cell, with inhibition of the TCA cycle and OXPHOS, production of lactate, and secretion of citrate. Next, we moved on lipid metabolism rewiring by MALAT1 depletion. Nuclear magnetic resonance is an attractive tool in metabolomics research. NMR spectrometry analysis show a strong downregulation of phosphocholine upon MALAT1 targeting, and also the quantitative enrichment analysis show a strong downregulation reduction of phosphatidylcholine biosynthesis paralleled by sphingolipid metabolism. The low level of phosphocholine prompts us to evaluate choline kinase A expression because it is responsible for the conversion of choline into phosphocholine. Time course analysis in androgen-independent and androgen-sensitive prostate cancer cells show a good downregulation of choline kinase A messenger RNA in MALAT1-depleted prostate cancer cell. We checked also sphingomyelin ceramide cycle. In addition to choline kinase A as a protumoral signaling, we focus on another protumoral signaling, that is sphingomyelin biosynthesis by these two enzymes, and also SERC activity that inhibits ceramide, promoting cell proliferation. Also here, time course analysis in a LNCAP cell show a good downregulation of enzyme involved in sphingomyelin ceramide cycle. Another layer of complexity is that choline kinase A and SERC are directly under the control of androgen plus androgen receptor, promoting choline kinase A expression and SERC repression. Those response curves show that choline kinase A is induced only at the higher concentration of androgen. Instead, SERC is repressed already at the lower concentration. To study MALAT1 mechanism of action in lipid metabolism rewiring, we want to study also the interaction of androgen receptor in androgen responsive element of choline kinase A and SERC gene upon MALAT1 depletion or androgen treatment. Of interest, androgen receptor is recruited in this regulatory region with the same extent of androgen treatment. This suggests that androgen receptor recruitment is facilitated upon MALAT1 depletion by Gapmer. To evaluate the interaction between androgen receptor and MALAT1, we performed RNA immunoprecipitation. We can observe a good association between MALAT1 and androgen receptor at basal condition. This association was abrogated after androgen treatment. So, MALAT1 targeting by Gapmer unveils a new liganded androgen receptor activity, repressing choline kinase A gene and SERC gene, promoting anti-tumor effect. To address MALAT1 as a possible target for RNA therapeutics, we used organotypic slice culture as a predictive model. We subdivided patients in non-recurrent and recurrent, and we combine classical histopathological and clinical parameters to MALAT1 expression and other metabolic gene expression. This new criterion, here I show you the single variable contribution to the final analysis. This new combined criterion show a better association with the progression of disease than classical criterion. Then we focus on MALAT1, on single host response to MALAT1 targeting, adding in this analysis also the target gene modulation by MALAT1 depletion. Also here, this new criterion show an excellent classification performance. Finally, we are developing a mathematical model able to predict a dynamic metabolic reaction system using metabolome time series data upon MALAT1 targeting in prostate cancer cell. Thank you for your attention. I want to thank all my colleagues from Università Cattolica del Sacro Cuore and CNR IASI.
Thank you very much. So, is there any question? Or we move to biotechnology. Now it's time to thank you very much for your talk. So Matteo Barberis, and so please. So I understand you're a biotechnologist, right? So it's very interesting.
Okay, so thank you very much for the invitation. Well, first of all, I would like to thank the organizers.
Just speak in the microphone, otherwise the recording doesn't.
I would like to thank the organizer of this exciting session, for the opportunity to present the work that we are doing at the University of Surrey in the UK. Okay, so the system, the biological system that we are working on is the cell cycle, the cell division cycle, eukaryotic one. So budding yeast, mammalian cells. As you might know, the cell cycle is divided in 4 different phases, as also we have heard from previous talks before. We have the growing phase that is happening in the G1 phase and in the G2 phases. And then we have the S phase where the DNA is replicated, and then the M phase where the DNA, where the cell divides. And within all this circle, essentially, you could ask a question: is a cell cycle a complex system or not? Well, if you look at the map, it's not quite complex as a metabolic map, but I would not consider this as a trivial either. So, indeed, it's a complex system, and you have many different interactions that interact and exploit a specific function in each of the different cell cycle phases. So G1, G1 to S transition, S phase, M phase, and so on and so forth. And interestingly, and very nicely, Professor Alberghina this morning, in her lecture, was stating this: it is often possible to identify a module which turns out to be an appropriate representation of a system as a whole. So this is what you would like to have each time that you address a biological complex system. And in this case, for the cell cycle, what you could do, you could either consider each phase as a module of this biological system, but you can also consider the actor that in the cell cycle are the cyclin-dependent kinase inhibitors as the module driving all the events through all the different cell cycle phases. This actually is what we are focused on. We would like to understand how the kinase complexes coordinate the cell cycle modules in the different cell cycle phases, because there is a property that is called waves of cyclins that is very much of interest, which drives all the different events through the different cell cycle phases, from the G1 to mitosis. And as you can see here, from human to budding yeast, this property is quite well conserved, which each of the cyclins rising and falling down at each specific time, one after the other one, as you can see both in mammalian cells and in budding yeast, but especially, particularly, decreasing or disappearing all roughly at the same time. So this is a very specific property of these kinase complexes. And our aim was to identify the molecular designs that were able to synchronize the timing of these cyclin waves, which basically means to understand which are the designs, if there are any, through which the cyclin CDK-dependent kinases coordinate DNA replication with cell division. So to set up with the experiments, with the pipeline for this work, we have first looked into the literature, and to look for specifically the transcriptional way in which all the different kinases might be coordinated or regulated. And as you can see, there is a large part of the cell cycle in which mechanisms through which specific cyclin-dependent kinase activities and transcription factors are very well regulating one another. And this is the main transitions between the end of the cell cycle and the G1 to S transition that also Marco Vanoni and Pasquale Palumbo mentioned in their previous talks. But as you can see here, there is a large part of the cell cycle in which there is a scattered knowledge about a few transcription factors, but not really kinase complexes activating or inhibiting them in a consistent way. Therefore, we decided to look into this in a bit more in detail, and through a systems biology pipeline integrating mathematical modeling and experiments, we did first generate a very simple mathematical model, including one main actor for each of the modules, so G1 module, S module, G2 module, and M phase module. This is the inhibitor SIC1 that is highly present in the G1 to S in the G1 phase, which is homologous of P27 of the KIP chip family in the mammalian cells. Then we took the CLIP5 and CLIP6 that are the cyclins driving the kinase in the S phase for the DNA replication, CLIP3 and CLIP4 driving spindle assembly in G1 phase, in G2 phase, and then CLIP1 and CLIP2 deputed to the cell division in M phase. And a very simple model in which we connected in a linear way all these three kinase complexes, and we look especially at the transcriptional activation or potential way in which these kinase complexes could be linked through this transcriptional activation. As to make a long story short, we build a number of different networks that miss or that lack either one or multiple of these potential transcriptional regulation, because I just recall you again that these transcriptional regulations were not known in the literature, and we try to reproduce qualitatively the waves of cycling behavior. So to make a long story short, we predicted from the molecular point of view that should be a molecule driving the connection between all these cyclings, and we validated experimentally using a wide range of experiments ranging from chromatin immunoprecipitation, protein-protein interactions, and so on and so forth. We discovered that there was a specific transcription factor molecule called FKH2, which is a highly homologous 4-cat transcription factors in eukaryotes, that was actually linking the three different kinase complexes in a linear way. But obviously, this is what I called design first, the design one of this network. This network that we have investigated is essentially one cell cycle from the beginning to the end, but does not repeat itself. But as we know, in all the cell cycle, all the transcription factors, all the kinase complexes, and all the other molecules, they come regularly in action. And there is no exception for this transcription factor that we have identified now being responsible for the link among the different kinase complexes, as there is a continuous interchange between the CDK activity through the 4-cat transcription factors and the transcription factor network of which FKH2 belongs to. Therefore, what we set out then was to try to identify networks, the designs that underlie not just one cell cycle, but a sustained oscillations to explain multiple cell cycles one after the other one. Therefore, we use the same mathematical model, but this time we include not only the transcriptional activation that is in red color on the slide, but also the post-transcriptional inhibition through which each of these kinase complexes is inhibited, as well as also we included the synthesis and degradation of the inhibitor SIC1, which in the previous model was just at the high level. And as you can see here, for simplicity, we called all the different kinase complexes as X, Y, and Z, and we explore all the possible combination through which the three different kinases will be able to generate oscillations. Essentially, what we will look for is a so-called limit cycle that essentially drives autonomously from the head to the tail of the cell cycle, from the beginning to the end. And what we want to observe is that our four components of the network, so the inhibitor for the G1 phase and each of the kinase complexes assigned to S phase, G2 phase, and M phase, come one after the other one, and we only consider the point in which these are fully sustained oscillations and not mild oscillation as at the beginning here. So I will just make a long story short. Whether in the previous effort we discovered the linear cascade through the molecule FKH2, here with these computational efforts, and for this we use a methodology called systems design space methodology, which has been used by Michael Savageau in the US, essentially for metabolic systems in the early times. We added to the linear cascade a few other interesting interactions that we have not seen before, such as a positive feedback loop on CLIP3, and also two negative regulations, which would make sense in the logic of the cell cycle, because whenever you are dividing, you would like to prevent any DNA replication of the DNA going on. Otherwise, you might have a not even distribution of the chromosomes at the cell division. So this was a computational prediction, which I would call the design 2. And therefore, you can see the difference or the similarity between the design 1, this linear cascade with a positive feedback loop on CLIP2, and the design 2, of which we have two variants in our system with or without the first part of the cascade. So you see that there is some part of similarity, but a lot of new things. And at this point, we wanted then to try to investigate the actual, what I call design tree, so might be the actual design that is in place in vivo. Therefore, we use these mathematical models to fit this mathematical model with the real cycling CDK dynamics. And to do that, we collaborate, one of the postdocs of our laboratory did go to Tartu in Estonia, and together with my friend Mart, they did generate time course data of the 3 different cyclings, one of the 3 different cell cycle phases, so S phase, G2 phase, and M phase, respectively red, blue, and green. And with Thierry, our PhD student, we have fitted with multiple models this time course data. We fitted both deterministically, specifically changing or fixing the value of 11 parameters and letting 10 parameters free of fitting, or stochastically inserting the, let's say, using an interval for the 10 structural parameters which connect all the 3 different cyclings, this X, Y, and Z, as you might remember before. So essentially, to make a long story short, we tested more than 21,000 fits for all the possible regulations among this different little network here that connects X, Y, and Z, the 3 different kinase complexes. And to avoid you to interpret these graphs, I just give you the solution of each of these graphs. So when we fit our models with the real data, what we do see is that indeed there is a linear cascade, but not only, there is also another branch that directly connects X to Z, means S phase to M phase, but you do see that the weight of this regulation is very different, predominantly with the last part of the cascade. And in addition to it, we also have, interestingly, as the strength of the regulations is higher not only for the last part of the cascade, but also for the 2 positive feedback loops for the clip tree, so in the middle of the cascade and also at the end of the cascade, as well as for the negative regulations, which are the anaphase promoting complex that are useful to degrade the cyclins at the end of the cell cycle. Nicely, these are as predicted, also as logic. Whenever you activate the cell division, you don't want the DNA replication and early events to act concomitantly. And instead, we don't have any role for the negative feedback loop on the APC cascade, let's say. So essentially here, what I tried to show until now is that we move from the different designs according to the using the model to predict an experimental molecule, which then we identified as a transcription factor able to drive this linear cascade of the kinase complexes, to a pure theoretical effort which was able to still predict that we would need a linear cascade among the 3 different cyclins to oscillate autonomously with additional deregulation of a positive feedback loop here and a couple of negative regulations to keep apart DNA replication and cell division. And finally, a fitting of our model to the real experimental data, to the time course data, showing that this linear cascade exists, but also both positive feedback loop exist at both levels on the different cycling levels. But here, to make it a little bit more interesting, we decided to use this model to predict some new feature of this scheme. So looking at our fitting of the model, we saw that essentially the last 2 cyclings of the cell cycle, so the G2 phase and the M phase, clip 3 and clip 2, were able to highly phosphorylate this transcription factor FKH2 as compared to the clip 5, to the S phase cyclins. And therefore, we decided to test this prediction in vitro, qualitatively, where we see that each of these individually purified kinase complexes increase the phosphorylation of FKH2, being the mitotic one more efficient, if you wish, as compared to the S phase one. But most importantly, in collaboration with Francesc Posas at the University of Pompeu Fabra, now at the ERB in Barcelona, we did test this in vivo, showing nicely that the early times, the phosphorylation of clip 5 and of clip 3, but mainly the one of clip 3, is strongly responsible for the phosphorylation of FKH2 and promptly getting to the S phase of the cell cycle and starting of the waves. Okay? So therefore, the quantitative model moving from the vision of the Nobel Prize Paul Nurse that first proposed this increasing of the kinase activity for the proper timing of cell division, here we discover that might be 3 different thresholds of S phase, of G2 phase, and M phase, obviously bound to CDK1, which in turn, one after the other one, phosphorylate the transcription factor FKH2, which in turn is the acting molecule to generate or to activate the next set of cyclins. And altogether, also taking inspiration with long discussion that we had together with Hans for almost 6 years when I was in Amsterdam, this model is sort of gear shifter for the cycling waves when you are connected, all the 3 of them with multiple mechanism, activation mechanism, brake mechanism, but then there is a specific turning control which biologically is driven by the axis between the fork head and the clip tree, which allows for the autonomous oscillations of the all molecules. And very interestingly, what I've been telling you for budding yeast can be also reconstructed very nicely for the human cells with a very similar interactors. And with this, I would like to conclude giving a few take messages from this talk. First of all, the complexity of dynamic systems can be addressed by looking at the simplified core models, that the complexity of a cycling cells is achieved through synchronicity to keep a precise timing of cell cycle progression. A principle of design that I presented synchronized the oscillations of mitotic cyclins to transcriptional regulation. And finally, the quantitative model of the CDK control is rationalized by specific regulatory motifs that keep a well-timed cycle. And with this, I would like to thank the many collaborators in the different projects, but on this one of the cell cycles, specifically Martin in Estonia, Francesc in Barcelona, Hans in Amsterdam, Edda in Berlin, and Ioannis in Switzerland. And thank you very much for your attention.
Thank you very much for your interesting talk. So I understand correctly that you don't want this talk to be broadcasted on YouTube?
Yeah, because of the collaborators. Yeah.
Is there any question? Urgent question? Okay. We still have...
Paul Nurse, of all people, told me, was telling us in a talk that one can delete clip 2 and still have a cell cycle. So my question is, could there be additional factors involved?
Thank you for the question. So actually the experiments of Paul Nurse, but also Mariano Barbasid in mice, they did delete all cyclins, but not clip 2. So the mitotic one is the one that is in there. And in fact, also in the different, if I manage to show it here, well, I can show it also here. In the cascade, in fact, everything obviously converges on clip 2 and also the majority of the regulations. That is possibly the reason why deleting the other one has a little effect on the viability also on the mice in mammalian cells, indeed. So there is a part of overlapping, but there is also part of specificity, but in the experiments clearly show that the overlapping part is much stronger for clip 2.
So thank you very much. Thank you again. Okay, so we still have the last 3 talks of the day. Thank you very much for being patient. I know there's not even a coffee break. I'm not responsible of this scheduling. Okay, so Anna Maria Colangelo. So she's going to talk about neuroscience, and she's from Milan. No, this is... Okay.
Sorry. Anyway, thanks for the
You said, let's hope that it works.
For the rush, the stress, and everything. So it's a pleasure to talk about my latest work done with Professors Alberghina and Hans Westerhoff and is about the modeling of mechanism underlying neuronal differentiation. So neuronal differentiation, and specifically the differentiation mediated by NGF. It doesn't work. So I was saying the modeling of mechanism involved in NGF differentiation. So NGF is the well, it's 1 week of Murphy's Law, so we are going on. Mi dispiace, più che altro perché siamo in ritardo.
He's coming.
Non lo so.
The technician is coming.
Vabbè, parlo senza... Is it okay? Anyway, I can start saying that NGF is a complex molecule in terms of structure and function, and it was discovered by Rita Levi-Montalcini in 1952 as a growth factor promoting the differentiation of sympathetic and sensory neurons. And so is the prototype member of the growth factor called neurotrophins, which promote the survival, the brain development and function. So I was saying that NGF promotes the differentiation of sympathetic and sensory neurons by regulating survival and differentiation and innervation of proper targets. And this occurs through two complex signaling. Now, in addition to this function, can you give me the next slide? And the discovery of the relevance of NGF in modulation of the neuroimmune system, also by Rita Levi-Montalcini, it was found that NGF is important for cholinergic neurons and so has a high therapeutic potential for Alzheimer. But recently we discovered a new function of NGF, that is the regulation of astrocytes activity and their role in controlling synaptic homeostasis. So if you can go to the next slides, this is an important property for proper function of the nervous system, which is based on the activity of 100 billion neurons, whose function is regulated by trillions of glial cells, and these through a number of functional neuroglial networks. Many of these mechanisms are not clear yet, as well the differentiation and proper target innervation by neurotrophic factors. So we applied a modeling approach to understand key elements directing neuronal differentiation. And we did this, next slide please, in collaboration with Professor Alberghina, the team of Professor Westerhoff, and in particular Evelina and Liu, and of course the people of my lab. Please go ahead. Okay, so for these studies we used the PC12 cells, which is a cell line derived from the neural crest, and so having the same embryologic origin as the sympathetic neurons, the sympathetic ganglia that you see on the left. And so in the presence of NGF, these cells, these tumor cells, differentiate into sympathetic-like neurons. And at the same time, they show an increase in mitochondrial membrane potential. If you can lower the lights, no, if you can lower the lights because you cannot see the, and also an increase in mitochondrial respiration. So all these observations brought to the demonstration that NGF differentiation involves a boosting of mitochondrial function to comply with the higher energy demand required by the differentiation. And this is important for brain development and adult neurogenesis. If you can please go ahead. So to better understand the link between NGF differentiation and mitochondria, we made a model, a mathematical model, centered on mitochondria and the three main processes regulating mitochondrial quality control. And so the model, that is, the design model is on the left, included ROS, which are produced under physiological conditions but are overproduced during a toxic stimuli compromising mitochondrial function, as well when mitochondria overwork to produce energy. And then it included fission-fusion cycling, which is the process regulating fragmentation of damaged mitochondria in the center, and the fusion of new mitochondria into cable-like structures. And the model, the design model, included two types, two classes of mitochondria, the networked and the dispersed one. ROS produced by networked and by dispersed mitochondria, the cycling between mitochondria, networked mitochondria and dispersed mitochondria, which was responsible for the passage of states between healthy, damaged mitochondria and severely damaged mitochondria, which are the ones that are going to be degraded by mitophagy. And plus, it considered also mitochondrial biogenesis. Now, the design model was translated into a mathematical model and used for simulations by the Copasi software. So the predictions of the modeling were used to plan new experiments on the right, aiming to assess ROS levels, as well morphology and mitophagy, and so to try validation of experiment of model prediction. Can you go ahead, please? So, okay, the Copasi simulations showed the strong relation between ROS, fission-fusion cycling, and mitophagy. In fact, increasing the fission rate, you can see on the left, caused a transient increase in ROS, red, an increase in dispersed damaged mitochondria in the lower graph, a decrease in network mitochondria, and also at the same time caused a transient increase of the autophagy flux on the right, increase of mitochondrial synthesis, and also of the fusion flux. On the other hand, when increasing mitophagy, you see in the center, the graph in the center, increasing mitophagy caused a decrease of ROS production by dispersed mitochondria and network mitochondria. Overall, this simulation showed also an increase of active mitochondria, as you can see in the graph on the left, and so an increase in the quality of mitochondria and leading to what is called mitopsis. Now, to validate the model, we first assessed the ROS level in the PC12 treated with NGF and found an almost complete overlap of experimental points with the model prediction you see in the graph on the right, except for the last point at 45 minutes. If you can change slides, please. And finally, we also assessed mitochondrial dynamic by live imaging of cells treated with NGF and then loaded with mitotracker green, which stains mitochondria in green, and then lysotracker red, which stains the lysosomes. So they're overlapping. When there is mitophagy, you will see yellow spots. And the quantitation, also the imaging that you see on the left, and the quantitation of mitochondria using a dedicated software that is able to do the segmentation of mitochondria based on morphology and other parameters like texture and so on, showed that there is an increase of dispersed mitochondria, the last graph on the top, increased dispersed mitochondria peaking at 3-4 hours, and also other two peaks at 20, at 26, at 24, 25 hours. These are time-lapse experiments imaging. The increase of dispersed mitochondria was paralleled by an increased overlap of mitotracker green and lysotracker, as you can see from the image with the yellow spots in the center. And this overlapping was increased during the first 6 hours in the perinuclear area, the middle in the graph. Instead, in neurites was observed throughout the time course that was done. At the same time, we found that during treatment with NGF, there was an increase in network mitochondria at 20 hours and 25 hours. These mechanisms were blocked by blocking autophagy with baphyllomycin and were not changed by an autophagy inducer like rapamycin. The increase of mitophagy is clear from the images, and I hope you can see the video showing also, if you can activate the video. No, it was the previous one. Can you go back to the previous? There is a video down there. Yes. No, it's impossible. Okay, anyway, doesn't matter. I hope the message is clear that our data confirmed, validated the model prediction that we had by Copasi. And if you can go to the last slide. So in conclusion, no, we skip this because I don't think. So in conclusion, next slide. So in conclusion, we show the construction of a model centered on mitochondria and the three main processes regulating quality control and function. The model has a good predictive value of the change induced by NGF differentiation, thus providing a valuable tool for model-driven experimental observation. And from this model, we also derived another model explaining the changes during NGF depletion as it occurs in aging and neurodegenerative disorders. And I cannot show, but all these mechanisms are shown in this cartoon. So I thank all of you for your patience and
thank you for your patience. So
well, this is the conclusion.
It happens once in your life. It happens to have
It's okay.
Slides stuck. Is there any question? Or I know we are very late, but we should be patient because still we have two talks that are. Thank you very much. Thank you. So on YouTube, we will remove all the problems. So now we have Alberto Bersani. Please. Sapienza University. Spero che funzioni. I hope everything will work. So about the asymptotic behavior of oscillating biochemical systems. So please, go ahead.
Thank you. I would like to show you some preliminary results on the stochastic approach to oscillating biochemical systems. The usual, no, please, no. Okay. The usual starting point is the Michaelis-Menten kinetics, where the second step is the usual standard quasi-steady state approximation that describes the binding between substrate and enzyme, which form a complex. But in our approach, we will use an alternative approach that is the so-called total quasi-steady state approximation, the TQSSA, which has shown to be much more valid in a much wider range of parameters and initial conditions, and has been studied for the last 3 decades. In this way, I am anticipating my first slides.
Please go with the second.
In our stochastic approach, we use the chemical master equation, that is the usual technique to study the stochastic phenomena. Clearly, when you consider a small number of molecules, you can expect that the stochastic approach is more useful than the deterministic one. And in fact, we, together with Gabriella Mavelli, Pasquale Palumbo, and Alessandro Borri, we started to study the stochastic counterpart of several mechanisms, in particular the so-called double phosphorylation-dephosphorylation cycle, obtaining quite interesting results. Okay, so we can skip, I suppose, very quickly. You can go ahead. This is a corollary of Murphy's laws. Okay, okay, you can go ahead. Okay. This is Michaelis-Menten, something like the rapid visits of Rome by buses. So you can go ahead. This is the quasi-steady state approximation. Here I will show in the next slide, I can show the so-called TQSSA, where we use similarly a quasi-steady state approximation, giving another set of equations. The next one, please. And as already I said, the TQSSA may be considered in some sense always valid. So the next slide. This is just a comparison between the application of the total approximation and the standard approximation in only one reaction. So we can go ahead, please. Okay, ahead. Okay, ahead. So here we are speaking about the chemical master equations that is based on a continuous time Markov chains. So you can go ahead. What was the reaction rates in the deterministic framework become the propensities in the stochastic one. So you can go ahead. Okay, we can go ahead. Ahead. These are all the mathematical part that probably we can skip. Okay, you can go ahead. So this is the chemical master equation in the case of our enzyme reactions. And we have ordinary differential equations in terms of the probabilities. so you can go ahead. And when we consider the matrix equation, we have to consider the stationary distribution of the probabilities. So we have to set equal to 0 the right-hand side part of the equation, so you can go ahead. Okay, these are all the mathematical properties of this matrix. Go ahead. Okay, so we can obtain the asymptotic distribution of probability distribution in terms of the matrix G. So you can go ahead. Okay, in general, in order to treat the stochastic, the chemical master equation, the commonly used algorithm is the Gillespie algorithm or stochastic simulation algorithm that is based on a larger number of simulations, and in some sense the distributions of the SSA tends to converges to the exact distribution. So you can go ahead. We applied our the stochastic approach to the so-called double phosphorylation dephosphorylation cycle. In the next slide you can see the scheme. Okay, and this scheme brings through the usual mass section law to the set of equations in the next slide. Okay, no, this is another way to show the scheme of reactions, so you can go ahead. Okay, and when we study the asymptotic distribution, the asymptotic states, we say that in the case of the double phosphorylation dephosphorylation cycle, we have a phenomenon, the bistability phenomenon. What is important is then when we apply the standard QSSA, we have absolutely a behavior completely different from what happens when you use the full system without any approximation. On the other hand, the usage of the total QSSA brings exactly the steady state states of the full system. In the next slide, we can see that we have some states where we have only an equilibrium point. In the next slide, we can see bistable points. Okay, these bistable points that are absolutely identical in the case of the full system and of the total quasi-steady state approximation. In the next slide, we can see what happens when we study by a stochastic point of view this bistable situation. When we study through the Gillespie algorithm the asymptotic distribution, we see that the distribution is bimodal and the two modes coincide completely with the two bistable states. So you can go to the next slide. What is important is that, let me recall that any quasi-steady state approximation is considered valid when we can see, in fact, a quasi-steady state. When you see, when you have to study cycles, you cannot in general expect that the approximation can be every time valid. In fact, when we see two particular cases where the total concentration of the enzyme changes only for one molecule, you have completely different situations in the sense that the full system and the total have bistability from a deterministic point of view and bimodality by a stochastic one. The standard approximation gives bistability but unimodality. When you pass to the next slide, changing only for one molecule, the total enzyme concentration, full system, standard and total give deterministic bistability and unimodality by a stochastic point of view. And we interpreted this phenomenon, you can go to the next slide, in the sense that one of the two modes, please the next one, one of the two modes is much more probable than the other one, and so it behaves like a black hole. Consider that when you study, apply any stochastic algorithm, you in some sense wait for an infinite time. So the system has all the time to switch from one state to another one. So it is in some sense natural to expect that the black hole attracts quite all the states. So please the next one. So then we asked this question: what happens when we pass to study oscillating systems? Again, we expect that any quasi-steady-state approximation cannot in general be valid. In fact, please the next one, in a previous paper with other collaborators, the next one please. Okay, the next again. We studied the so-called mitogen activated protein kinase cascade with feedback, and our results were that, please the next one, that the approximations, the total or the standard, were in some cases were able to reproduce the oscillations of the system, but in other cases there were completely different situations. The full system gave steady state, but the approximations gave oscillations, or vice versa. So every time we apply any quasi-steady approximation, we have to consider that cycles, oscillations cannot guarantee the validity of the approximation. So we passed to study a simple oscillating mechanism, the next one, the so-called per cycle, studied by Goldbetter even in 90s. And in this case, Goldbetter showed oscillations like limit cycles and so on. In the next one, we describe the reaction. This is the full system. We rebuilt the full system because Goldbetter directly applies the standard approximation. The transcription, the nuclear transcription is treated by an Hill function. The degradations are treated by Michaelis-Menten, and we conserved these terms, as you can see in the next slide. Okay, this is the full system of the per cycle. The Hill functions are represented with the parameter equal to 4. So the next one, this is the stochastic counterpart. The next one, this is the standard approximation. All the derivatives of the complexes are set equal to 0. You obtain the equations. In the next one, you can see the stochastic counterpart. Then we applied also the total quasi-steady-state approximation. Consider that the total concentration of P is not conserved. We introduced, as usual, the total concentrations. These are the equations. Then in the next slide, we can see where the approximation applies. In the next one, we can see equations of the total approximation. Then we applied the three systems and obtained results. What is in the next slide? What is curious? Okay, this is the counterpart, stochastic counterpart. In the next one, okay, at last results. Okay, the next one. Okay, curiously, Goldbetter misses in figure 2 the initial conditions for M, for the mRNA, and for P. So we really, with rule and with a lens, try to detect these initial conditions from figure 2. We treated figure 2 and figure 3. Okay, the next one. These are the initial conditions. Okay, the next one. Okay, as I told you, we didn't find the initial conditions for the for the mRNA. The next one. Okay, this is for figure 3. In this case, we have the initial conditions for M and for pair. Okay, the next one. At this point, we simulated the two figures. This is the full system, and in the next slide we can see the comparison with this is the full system. This is the phase space for the full system, where you set the concentration of pair, the total concentration of pair and mRNA. Okay, this is full system. The next one, this is TQSSA in the next, and this is standard QSSA. As you can see, there is a big difference. Very rapidly now the other figure. Okay, the next one, this is the total. Sorry, this is the full, this is the total. The next one, this is the standard. Again, another situation. This is the full, this is the total, this is the standard. So it seems it was quite astonishing that the total could still approximate in a very well way the full system. Okay, very rapidly the next one. Okay, what we expect, and this is the last thing I want to say, is that the distribution of probability is something like the famous higher law. I mean, in a cycle, I suppose that every state is equally probable. But when we studied the stochastic asymptotic distribution, please the next one, we obtained something very curious. This is the distribution of mRNA, and we have a bimodality for mRNA. The next one, we have even unimodality for the total concentration of pair. So it was a bit surprising for us. Okay, let us skip to the next one. I suppose that they are okay. These are the joint distribution for pair and for mRNA. Okay, the next one again. This is the level curves, and we see something like a banana-like shape for the joint distribution, and this is still a subject of our studies. I think that we can pass to conclusions at last. Please, the next one, okay, you can skip, okay, conclusions at last. So, for sure, it seems that total approximation is much more valid than the standard approximation. What we want to study is to understand, by means of the total that simplifies, obviously, the stochastic framework approach, and by means of the full system in the next one, what we obtained in the stationary distribution. Okay, you can skip. We are at this point, the next one, at this point absolutely in a situation of a work in progress. We cannot give in this moment any answer. So the last one, our future aims are to interpret the modality of the case of pair cycle, to understand what happens when you increase the number of molecules in order to see if the stochasticity is lost, and what happens in general to this distribution also in other cycles that we are going to study. I think that this is the last one, please. No, okay. This is only an answer related to the banana shape. It seems that the two concentrations of the total P and the mRNA are in some sense correlated, so probably the banana shape is related to this experimentally seen correlation. So I think that it is the last one. Okay, you're sorry for.
Thank you very much, and sorry for all the technical problems that interrupt the exposition. So I think we are super late. I'm very sorry, but so we have our last hero, Alessandro Borri, who was. Grazie. So Alessandro is from the CNR, and he's going to talk about model and control of the glucose insulin system.
Good afternoon, and so next. I am Alessandro Borri from CNR Biomedics Laboratory. This is glucose insulin is a research line in collaboration with the people that you see in this slide, mainly belonging to the CNR and the University of L'Aquila. Next. So the glucose insulin feedback loop is an example of complex system in biomedicine whose importance most lies in the fact that glucose dysregulation leads to diabetes. To tackle the problems of glucose prediction and regulation, we need good models of glucose insulin system. Next. This is an example of natural regulation in biology where endogenous insulin is secreted by pancreatic beta cells in response to circulating glucose. Next. This leads to formalize a two-node negative feedback loop in which glucose promotes the production, the release of insulin, and insulin represses glucose. Next. When this regulatory system doesn't work, we have diabetes, which can result from defects in insulin secretion, action, or both. We can have type 1 or type 2 diabetes. Next. Diabetes reached the epidemic proportion in the last few years, thanks to mostly because of the lifestyle and in Western society. Next, this is often associated to metabolic disorder, including some other risk factors such as blood pressure, high blood pressure, and excess body fat. Next. Fortunately, in the last few years, technology made possible exogenous regulation by means of an artificial pancreas system. Next. So the challenges in glucose control come from the two dangerous conditions of hyperglycemia and hypoglycemia. While long-term risk of diabetes are caused by persistent hyperglycemia, which causes chronic issues such as microvascular damage, renal insufficiency, and neuropathy. Next. Short-term risks are associated to an excessive dosage that you can have in therapy. So if you give too much insulin, since insulin clears sugar from the bloodstream and most organs and tissues work on sugar, this can lead to severe negative effects that in worst cases can also lead to death. Next. So the final goal is to stay within a very narrow interval of glycemia. Next. Models of glucose-insulin system can be divided into maximal or extended model, which are good from the prediction viewpoint, but not very useful from the control design viewpoint, in which we typically need algebraic or differential manipulation. Instead, reduced order models have limited prediction capabilities, but can be used as a basis for a control design. In the middle, we have some medium order models. Next. Examples of high-dimensional models usually include in the glucose-insulin picture some additional players such as hormones, food intake, glucagon, and subcutaneous compartment. Next, other models deal on the microscopic secretion by the insulin firing units in our pancreas. This is a stochastic behavior, but since we have an aggregate behavior by hundred thousands of firing units that act independently, the macroscopic behavior can be approximated by a deterministic medium order system. Next. For control, we mainly use compact models. It's worth to mention that not all the control approaches are made on models. Some of them are model-free and are mainly adjust parameters based on human expertise on experimental data, but they are typically less accurate than model-based controller. Next. So around 15 years ago, there was a research effort at CNR to design an advanced model to overcome some issues from previous ordinary differential equation models, which could not represent in an adequate way some phenomena related to the glucose response, for example, the second release of insulin. Next. This led to the design of this SDM model by Panunzi, Palumbo, and De Gaetano. This is a model with two variables of interest, glycemia and insulinemia, but because of the presence of a discrete delay in the equation, this is a delay differential equation model, so it has an infinite dimension. Next. This model has some nice properties. For example, it has a single local attractive equilibrium, positive and bounded solution, and statistically robust. And is a compact model in the sense that it depends on a minimal set of physiological parameters, which can be effectively estimated with simple experiments. Next, last but not least, it provides a nice fitting of experimental data. Next. In the second part of the talk, I will discuss some control design techniques based on the SDM model. In the first part, I will discuss event trigger stabilization. Next, in the second part, I will talk about formal methods. Okay, go ahead. Next, okay. Since the DD is an infinite dimensional model, we need to approximate the infinite dimensional state space, and we exploit splines, which are piecewise linear function. It is possible to show that with a finite number of splines, it's possible to approximate with arbitrarily good accuracy a continuously infinite number of space of Lipschitz continuous functions. Next. So, what is the event trigger paradigm? In digital control, usually tasks are executed periodically. Next, instead, in the event trigger control, to save computational resources, we control the system whenever it really needs attention. This is similar, to use a metaphor, to a wise man who speaks because it has something to say and not just because he has to say something. Next. Okay, sample data control is made of three basic steps. The first one is the design of a feedback law in continuous time, achieving asymptotic stabilization of the error. Then we sample and hold the control law to obtain a piecewise linear function, and this preserves stability in a practical sense. Finally, the event trigger mechanism consists in the fact that at each sampling time we check a triggering condition and we update the control law only if the current input is not expected to guarantee sufficiently good behavior in the near future. Next. Simulations show that in glucose control, if you reduce the number of control updates to just 20% with respect to the periodic solution, you have an almost undistinguishable behavior with respect to the time trigger control by the event trigger control. Next. So finally, I talk about formal methods, which are a framework developed in the context of verification on software and hardware systems and allows to put together heterogeneous systems, in particular discrete and continuous one. Next. In biomedicine, this led to the concept of a cyber-medical system, which is a digital system including mathematical algorithms that are tailored on the patient's physiology, control engineering, artificial intelligence, and big data. Next. Formal methods allow to manage complex specification, which can be thought as an arbitrary sequence of elementary logic predicates as safety, reachability, and avoidance. These specifications can be turned into language specification. To explain what is a language specification, think of a trajectory that is sampled at each sample time and you get a finite number of output symbols. If you put together this sequence of symbols, you obtain a word. A language is simply a collection of good words that can be encode desirable trajectories. Next, to give an example, if you are in an abstract space and you want to start, you need to start from an init state space and set, and reach a target set by avoiding an obstacle. Next, what you can do is to partition, to grid the space in order to turn the infinite set of states in a discrete finite one. Then, next, you can cover the obstacle and you can consider a subset of the initial and final set. So at this point, if you associate a yellow, red, and green symbols to init, obstacle, and target state, you have the specification, so the collection of good, of nice trajectories is the one, contains the words starting with yellow, ending with green, and we know red symbols. Next. Logic specification can be expressed by finite state machines. Next. And this is the framework that we employ, which is the transition system, which is made of a collection of states, labels, transitions, and outputs. Next. It's obvious to rephrase a purely discrete system in the form of transition system, for example, a slot machine or a coffee maker, but next. It's not very well known that you can also take a continuous time system and turn it into and have a finite approximation in terms of transition system. In order to do that, what you can do is consider all trajectories that start from a cell of the grid, and then next, you consider the evolution at some sampling time. Next. And then you go into a target set, you partition the target set, next, and so you can encode all the possible trajectories, next, in a finite set of transitions. Okay? Go ahead, please. The approximate equivalence, because original and finite system can be made possible by an approximate equivalence notion, which is called by simulation, which was called a bridge between computer science and control theory. Next, in the construction of this finite model, which are called also a digital framework, first we started by designing a controller satisfying a time-varying glucose specification during a day based on an average patient model. Then what we did was to test the robustness of this algorithm by building a population of 10,000 virtual patients, each patient generated by a random perturbation of the nominal parameters. And what we got next is that we were able to keep all the population very far from the dangerous E zone, the red one that you see in the top right-hand corner of the screen, which is representative of many hyperglycemic and hypoglycemic episodes. Next. As an ongoing work, I just mentioned that we are trying to add to the ordinary and delay differential equation modeling some fractional and stochastic terms, accounting for model uncertainties and mixtures of unknown phenomena, unknown mixtures of phenomena of different orders that may affect the variable of interest. Next. In the context of formal methods, we are investigating the contract-based design. Contracts are a recent way of encoding conditional specifications. Thanks to some compositionality results, we are able to satisfy contracts on very large systems by just guaranteeing the satisfaction of the specification on the components. Next. I leave you just some slides, slide with some further related and ongoing work, and I thank you very much for your attention and patience.
Thank you very, very much. Thank you to everybody for being so patient. It has been a bit of a brainstorming. I hope that some stimulus came out. And so thank you very much for coming, and everything will be broadcast and you will be asked for permission and etc. So have a good evening, and thank you very much for coming. Bye-bye.